In one of the previous lectures, we discussed Agrobacterium tumefaciens, which basically infects trees, especially dicotyledonous plants. And now, what sort of secretory pathway it uses to basically infect those plants? So the pathway it uses is known as type 4 secretory pathway. Let's look this particular pathway in a bit more detail. TI plasmids contain two sort of operons. One is known as Ver-B and one is Ver-D. They comprise about 11 and 5 genes respectively. Ver-B function as a part of the main secretion apparatus, which basically ultimately forms the secretion apparatus. And then Ver-D, which consists of 4 genes that encode 4 proteins, Ver-D 1, 2, 3, and 5, involved in the processing of the tDNA. Now this tDNA is known as transfer DNA in the TI plasmid. And one Ver-D4 which couples to the tDNA and the secretory protein. So you have to be very cautious. Any nuclear localization signal, anything which is encoded in DNA actually cannot function on its own. Any bodily reaction, anything happening inside the cell works or identifies various things through some signals, through some sort of interaction. For example, if I'm talking about nuclear localization signals, if I'm talking about something, a receptor which is going to change its conformation, it has to interact with some other protein, and that will be through charges. So if you have a lot of charged residues, that would be a localization signal, but you will have to see the sequence. So if the DNA has to be transferred, still there needs to be some proteins which are going to modulate that transfer. Now, Ver-B4 and Ver-B11 both contain nucleotide binding domains. Why they contain nucleotide binding domains, I don't have to tell you again and again, because ultimately it's a translocase and it is going to bind to bind to ATP or GTPs to ultimately draw the energy to do the reaction. So basically it's very simple that they provide energy for the translocation of proteins through the secretion of Ver-D4 proteins, which is also known as Walker domain, which is a nucleotide binding motif. Walker is coming from Professor John Walker, who works at University of Cambridge. He won the Nobel Prize for finding out the structure of ATPase, that how ATPase works to produce adenosine triphosphate, which is the energy currency of your body. Now you guys are in advanced courses. Also, you can find a very good experimental manual and a book on the structure of protein by 2 Cambridge scientists. One is John Walker. It's known as Protein Protocols, and the other book is by Sir Alan First on the protein folding. Okay. Of the remaining proteins, Ver-B6, Ver-B8, and Ver-B10 all lie in the inner membrane. So at the moment, we don't know the precise function of these 3 proteins, but they all show a propensity to interact with several other members of the secretion system. So let's suppose if I'm talking about at the protein level, if I don't know the function of a protein, usually we use protein-protein interactions to probe that structure. For example, there is this cascade. We know that there are 5 proteins. We know the end function of the last protein, or we know the function of the first protein. Then we see that what other proteins these 2 proteins are interacting with. And if they are making a lot of interaction, then we can start to guess that these intermediary proteins might be functioning this. They might be transferring an electron or whatever function they are playing. And the technology, the techniques we use for these is things like fluorescence, resonance energy transfer. We'll come to that in some of the next lectures, and the lectures the course where you would learn about the experimental techniques. Okay. So, and basically what we get to know through these interactions is that it's a suggestion that there is the interaction between these proteins and they are the part of the whole component. So if suppose we have a whole complex and we keep on finding that there are these proteins which are involved in that complex, then they must have some sort of role. This particular type 4 secretory pathway is not only limited to dicotyledonous plants. Bordetella produces pertussis toxin, which causes whooping cough. It is an exotoxin. So what do we mean by exotoxin here? It needs to be secreted out of the bacteria. So it means that type 4 secretory pathway can throw out basically huge proteins as well. Helicobacter pylori, Helicobacter pylori codes for a protein known as CAG-PAI. This bacteria is a beauty in its own right. If you look this bacteria live under the microscope, the way it rotates, some of you might just fall in love with this particular bacteria, the way it is continuously rotating. But this bacteria pretty much digs hole in your epithelial walls. So this protein produces bacteria. For example, it produces a sort of, you can say, a toxin, which is peptidoglycan in nature, which turns on the cytokine pathway for inflammation. तो जैसे ही एपिथेलियल सेल्स में टॉक्सिन जाता है, एक इन्फ्लेमेशन का कैस्केड है जिसको साइटोकाइन पाथवे कहते हैं, वो एक्टिवेट हो जाता है. You will learn about this cytokine pathway from the courses in immunology. This is a very good image because it shows you an image that how Helicobacter pylori is basically breaks up the cell wall, your mucous layer of the stomach. Basically, it disrupts the cytoskeleton and adherence to adjacent cell, thus disrupting the inner lining of the stomach. And if you look at the clinical studies, it's a huge burden. You can see two images here which are gruesome, that you can see a hole in the inside lining of the stomach wall and you can see a perforation again in the duodenal lining. So you can see that these secretory pathways are very, very important. I'm a bit sad that mostly courses in Pakistan ignore these pathways altogether. But to understanding them and ultimately either designing drugs against them or to use them so that we can excrete proteins of interest out of the cell have huge biotechnological significance.